a nightmare.
Activate.
Alright guys, so what we got here is the Biology Higher Tier Paper 1 2019. So we're going to be going through these questions and answers, and we're going to discuss them and try and figure out why the answers are correct, and if there's any side notes to write down, we'll tell you about it, and then we're going to get this done. Okay, right. So let's start with question number 1. Okay, so it says figure 1 shows an animal cell view using a microscope. The question says, the cell contains a nucleus. What is the function of the nucleus? Well, the function of the nucleus essentially will be to control the activities of the cell. Any of the chemical processes are controlled by the control center, which is the nucleus. Okay, so your answer will be controls the activities of the cells. And that's your nucleus right there. Question number 1.2. Name one type of cell that does not contain a nucleus. Well, inside the human body will be the red blood cells, and they don't have a nucleus, so they can make more space to carry more oxygen at the same time. Now, another examples could be bacteria or prokaryotes, because they do not have a nucleus, and you could also mention xylem in plant cells, okay? But just to be safe, and the one that is a lot easier to remember will be the red blood cells, okay? So your answer could be any of the following. Okay, so we got question 1.3. Draw a simple diagram of the cell in figure 1 and show 2 of the parts of the cell. So it's a 2-mark question, so you don't have to go into too much detail. It's basically what it tells you, okay? Now, you will have, first of all, your cell. That will be your cell membrane. And I'm just going to draw all of the parts that we normally be asked about, just in case you have any preferences, okay? So you got cell membrane. Right, so what we got here, right, so what we got here is the diagram of the cell, and we have the nucleus, mitochondria, ribosome, cell membrane, and cytoplasm. And the reason why we're not doing the plant one is because in the previous diagram, you can't see a permanent vacuole and you can't see a cell membrane. Cell wall. That's what it means. The cell that it was drawn in question number 1, it was actually an animal cell. So then it says here, name one structure found in a plant cell not found in an animal cell. Well, I just mentioned those, and it could be any of the 3: chloroplast, cell wall, and a permanent vacuole. So there you have it. Right, so let's move on to question number 5. It says here, figure number 2, sum of the different cells, and then you have their length, the real length from point X to point Y is 0.5 millimeters. So let's keep an eye on that, and let's pay attention to the units over here, okay? Calculate the magnification and use the equation below. Okay, so to calculate the magnification, you're going to need the real size of the image and the size of the image, and also the real size of the object. Now over here is telling us the real length of the object, so that there will be here, and then the size of the image, you have to measure the image size yourself, okay? So let's see what we can get with that. So we're gonna put the ruler from that line to this line, and if we take note of the units at the bottom, it says millimeters, so we're gonna count in millimeters, okay? It's very important that you remember that. So we got 10 millimeters, 20, 21, 22, 23, 23 millimeters, okay? So that distance there will be 23 millimeters. Now that we got that measurement, we're gonna go back over here to the equation, and then just to be able to remember these equations when you're practicing for your exams, it's always good to write down the whole equation, okay? I always tell my students that. Most of them never listen, and then we got issues because they don't remember the formulas. If you're doing a triple higher, normally sometimes you won't even get the equation, so you just gotta remember that by yourself. So it depends on the question. So let's go for magnification. Okay, so now that we've done that, in the calculator, let's type in 33 divided by 0.06 equals to 383.33. Now, there is no magnification at 383.33, so we can assume that the closest whole number that we could have there, it would be 400. So you can round that up to 400. So that's being 400 times magnification. Now, when I measured it, it measured to me 23 millimeters. The mark scheme actually says 24 millimeters, and when you measure 24 divided by 0.06, it gives you exactly 400. Now, the mark scheme actually says that you can have a range in between measurements because of the printing size. You could measure it wrongly, but you still get a mark if it's within the range of 23 millimeters to 35 millimeters. I think that's pretty big range, but still that's good, and that's the way you do. Okay, you have the real size of the object that is given to you, and then the size of the image is what you measure. Divide one by the other, and then you get your answer. Okay. Question 1.6. The cells shown in figure 2 were viewed using a light microscope. Give 2 advantages of using an electron microscope instead of a light microscope. So the advantages that you want to write are for the electron microscope, not for the light microscope. Okay, so what are the advantages that we have with the electron microscope? First of all, we got 2 types of electron microscope. You got the scanning and the transmission electron microscope. Okay, but you don't want to get too into much detail with that. We're going to go with the simplest things, which is the electron microscope gives you a higher magnification and it gives you a higher resolution. Okay, and there you have it. Those are your 2 marks over there. Okay, so we're going to move on to question 2.1 in the next video. Click on the next video and I'll see you then.
It was a nightmare. Activate.